Okay, so we've talked about a survey of prokaryotic cells and microorganisms. Now it's time to do the same thing, but with eukaryotes. So what makes a eukaryote a eukaryote? Well, what's special about us is we have metabolic processes that are located within membrane-bound organelles. These would be things like the nucleus, mitochondria, the Golgi apparatus, etc. And we'll talk about each one of those in detail here in just a little bit. How did eukaryotes evolve? Well, big picture, they evolved from prokaryotes to eukaryotes, just single-celled organisms. And then eventually those single-celled organisms became multicellular. Now, how did the first eukaryotic cell evolve? That's going to be endosymbiotic theory. This happened when a large prokaryote engulfed a smaller prokaryote, and that smaller cell learned how to live inside of the larger cell, essentially. And I'll talk a little bit more about that in just a moment. Multicellular eukaryotes evolved once individual cells started to lose the ability to survive independently. So you have trillions of cells in your body, about 40 trillion, and they all are grouped into different tissues that perform different functions. Then you've got groups of tissues that make up an organ. And each organ is going to have its own function that it performs. And then you've got various organs that together perform a common task. Those are going to be organ systems. So we are multicellular, and you can see kind of how the complexity builds. And all of this is because somewhere along the way, some individual cells lost their ability to survive without some other individual cells helping out. And so that's the, in a nutshell, the process of evolution to multicellular organisms. Okay, so let's talk a little bit more about endosymbiotic theory. So we'll start here with the proto-eukaryote, right? So this is thought to have been a large prokaryote. And early on, it starts to have infoldings of the cell membrane, and these infoldings eventually become the envelope around the nucleus, as well as the membranes forming the endoplasmic reticulum. Now, this proto-eukaryote is thought to have, at this point, engulfed another prokaryote, probably an aerobic bacterium, and eventually this aerobic bacterium learns how to survive inside of the cell to the point that it cannot live without the other prokaryotic cell. And that's the lineage that is believed to have become the mitochondrion. And one of the reasons we think mitochondria came from aerobic bacteria is because mitochondria use oxygen to produce ATP, and so do aerobic bacteria. Now, some of these early eukaryotes went on to a second endosymbiotic event where a photosynthetic bacterium is consumed in addition to the aerobic bacterium. And these photosynthetic bacteria evolve to the point that they cannot survive independently of this prokaryote, and eventually evolved to photosynthetic eukaryotes like plants. These guys down here, the ones that did not have that second endosymbiotic event with the photosynthetic bacteria, they go on to become modern heterotrophic eukaryotes. So that would be organisms like the protozoa, fungi, and animals. These are the eukaryotic organisms we're going to talk about over this course. The protozoa, these are unicellular eukaryotes. They lack a cell wall, and some of them have flagella or cilia that help them move about in their environment. Some of them are amoebas that have pseudopods or false feet that help them move around their environment. Then you've got the fungi and the algae. Fungi being can be unicellular or multicellular, as well as colonial. Fungi has a cell wall. We'll talk about what makes fungi's cell wall unique. Algae. This would be another example of photosynthetic eukaryote. And again, algae can be unicellular, colonial, or multicellular. And then these guys we'll talk about a little bit. Helminths, the parasitic worms. Lots of interesting parasitic worm infections that affect humans. Those are multicellular except for the reproductive stages, so things like the eggs of the worms, might be single cell, but the organism itself eventually develops to be multicellular. And we'll touch a little bit on arthropods, just because they happen to be vectors of disease. They are an animal that can carry a disease from one place to another. All right, this is a graphic just kind of pointing out the different anatomical parts of the eukaryotic cell that we'll be talking about. You can see up here we have a list of things that are found in all eukaryotes. Then down here there's a much shorter list of things only found in some eukaryotes. All right, so again, let's work our way inwards. So we'll start with the outermost structures, and these are going to be the appendages of locomotion. So the flagellum and the cilium. So the flagellum is a long sheath cylinder containing microtubules in a 9+2 arrangement, and I'll show you what that looks like here in just a minute. It is used primarily for locomotion. Unlike the one that we saw in prokaryotes, the eukaryotic flagellum is going to move in a whip-like fashion, okay? It's also going to be covered by an extension of the cell membrane. I already mentioned it has microtubules in it, and it's thicker than prokaryotic flagella, 10 times thicker, in fact. Now, cilia are going to be similar to flagella, but they're shorter and more numerous, and you're only going to find them in a few different protozoa and in certain animal cells. What's really different about them is they move in a coordinated beating motion. They're kind of like these finger-like projections that sweep substrate past the cell. So for example, your respiratory epithelium, what they do is they move mucus up out of your lungs. And the point of that mucus is to trap dirt, dust, and microorganisms. So the microorganisms, you breathe in, you're breathing in the dirt, the dust, it gets trapped in the mucus, and then the cilia of your respiratory epithelium move that mucus up out of your trachea, and you cough it up. And then that way it doesn't sit in your lungs and cause an infection. That's just one example of cilia. This is a cross-section of a flagellum, of a eukaryotic flagellum, and we know it's eukaryotic because of this 9+2 arrangement of microtubules. And what that means is that there are 9 couplets, so you can see 1 microtubule, 2 microtubule, and they're kind of attached. So this is a 1 couplet. There's 9 of these around here, and in the center there are 2 separate microtubules. You can see that they are not touching. That's all this 9+2 arrangement means. Okay, so that was appendages. Next, we're going to talk about the glycocalyx. And this is the outermost boundary of the cell that's going to come into direct contact with the environment. Usually it's composed of sugars, polysaccharides. It's going to appear as a network of fibers or a slime layer or a capsule. Its functions are to help the cells stick to things, to protect the cells from drying out, and to help receive signals from the environment. What is beneath the glycocalyx? Well, fungi and most algae are going to have a thick, rigid cell wall that protects them. Protozoa, a few algae, and every animal cell is going to lack a cell wall. So these guys have only a cell membrane surrounding them. Which brings us to our next structure, which is the cell wall. In eukaryotes, just like in prokaryotes, it's going to be rigid and provide structural support and shape. Now, we learned that bacteria have a cell wall made out of peptidoglycan. Fungi have a cell wall that's composed primarily of chitin or cellulose, but the one I want you to remember is chitin. Algae, don't worry too much about this. Just know that it can vary in composition, just depends on the species of algae. Okay, this is an image of the fungal cell wall. So let's start on the inside of the cell, which is right here. This would be the cell membrane. And then moving further outward, you've got a layer of the cell wall that is chitin, a layer of the cell wall that is glucans or sugar molecules, and a layer of the cell wall that is manoproteins. Okay, the cell membrane or the cytoplasmic membrane. This, just like in the prokaryotes, is going to be a bilayer of phospholipids with those proteins kind of floating around in those phospholipids. Now, in eukaryotes, sterols are going to be important. These help confer stability to the cytoplasmic membrane, particularly cholesterol. The cell membrane is going to be selectively permeable, and it's going to function in interaction and surface adhesion, secretion, and sending and receiving signals. Okay, so here is a diagram of the cell membrane. This is the extracellular space up here, so this is outside of the cell, and this is the cytosol, right, inside of the cell. You can see the phospholipid bilayer. So you've got the hydrophilic or water-loving heads that are pointed towards the fluid of the cytosol and the fluid of the extracellular space. And you've got those hydrophobic tails pointing inwards towards each other, trying to avoid interaction with the water. Then you've got proteins that are embedded in this phospholipid bilayer, and they kind of move around, just sort of floating among the phospholipids. Notice here is a cholesterol molecule, and you'll see them interspersed throughout the plasma membrane. What these guys do is they help kind of they prevent the phospholipid bilayer from being too flexible, too fluid. They give it a little bit of rigidity, a little bit of stability, kind of preventing these proteins from floating all over the place. And you'll learn that most prokaryotes do not use sterols. They don't need to because they have a cell wall. However, there is a type of prokaryote that lacks a cell wall, the mycoplasma bacteria. It instead uses sterols in its cell membrane to give itself extra stability and rigidity. So it kind of adapts almost a eukaryotic strategy to give its cell membrane strength. Okay, on to internal structures. So this is a list of the organelles we're going to talk about and their functions, okay? This chart is actually really useful for studying So I would refer to it frequently when you're making your notes, your flashcards, however you like to study. This is a good chart for studying the eukaryotic organelles, and there will be questions over these. So let's start with the nucleus. The nucleus is going to be a compact sphere, and it's also the most obvious organelle. It's the largest organelle of the eukaryotic cell. It's going to be surrounded by a nuclear envelope, and this is essentially like a double membrane with a space in between it, and it's perforated with pores to allow large molecules in and out, such as RNA. The nucleus contains the chromosomes of the cell, so the DNA of the eukaryote. Within the nucleus is a dark staining area called the nucleolus. This is the site of rRNA synthesis and ribosome assembly. rRNA, the R there stands for ribosomal RNA. So ribosomes, remember, are going to be made up of this ribosomal RNA and proteins. And so the nucleolus is going to be the site where some of these ribosomes are put together, parts of it. All right, so here's an electron micrograph showing the nuclear envelope, and you can kind of see there's two layers to this envelope. Here is a nuclear pore, and this is where things like the RNA would come out, mRNA to be translated. You have this dark staining kind of fuzzy stuff here. This is chromatin. This is the genetic material inside the nucleus. And then here's this large dark area, the nucleolus, the site of ribosome assembly. And then I just wanted to show you this picture real quick because it's got kind of a better or more clear illustration of the double membrane surrounding the nucleus. So you can see here is one part of the membrane and it kind of folds back on itself, the other part, and throughout it are these pores. Next you have the endoplasmic reticulum, which is kind of like this interconnected network of membranes. There's two types of endoplasmic reticulum. There's the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum appears rough because there's a whole bunch of ribosomes embedded in its membrane. So as you might guess, if you remember, ribosomes are the site of protein synthesis, so you may guess that the rough endoplasmic reticulum is involved in protein synthesis, packaging, and transport as well. The smooth endoplasmic reticulum, on the other hand, lacks ribosomes. It looks smooth because there's not a ribosome in sight. Instead, the smooth endoplasmic reticulum is going to function in things like storage of lipids, also detoxification, especially in cells like liver cells. Okay, so here you can see is the nucleus. This is the nuclear membrane here, the nuclear envelope. And then these are pores, the nuclear pores. And then you see that the pores kind of join into this network of membranes. And there's a portion of the membranes that have ribosomes in it, and then a portion of the membranes that don't have ribosomes in it. And then if you zoomed in on the rough endoplasmic reticulum here, the portion that has ribosomes, and you looked really closely, you would see here is a ribosome, right? You have the small subunit and the large subunit. And coming off of here is the protein that's being synthesized. And it's being synthesized from this strand of mRNA. This process, you may recall, is called translation. That's where you take an RNA transcript, such as mRNA, and you turn it into a protein, essentially. You make a protein using the RNA instructions for it. Okay, where do those proteins go once they leave the rough endoplasmic reticulum? They have to go somewhere. Well, the next organelle that these proteins are going to go to is the Golgi apparatus. And what the Golgi apparatus does, I want you to know, is it modifies these proteins that come to it, and then it ships them out. And that's essentially all you need to know about the Golgi apparatus. You can see it's kind of this funny looking stack of flattened sacs, and these are called cisternae. And you've got two faces. You have the cis face and the trans face. And what happens is the rough endoplasmic reticulum's somewhere over here, and pieces of the membrane of the rough endoplasmic reticulum bud off and become transport vesicles. And then these transport vesicles are full of those proteins that have been made in the rough endoplasmic reticulum. They come to the cis face of the Golgi apparatus, and then they just kind of merge into the membrane of the Golgi. And within these cisternae is where the proteins are going to be modified. And they're modified just by adding different chemicals onto them. Once all of that is done, you come over here to the transface, and you can see the membrane of the Golgi here just kind of buds off. And inside of it is the proteins that have been modified by the Golgi, and they're ready to be shipped to organelles or outside of the cell. Here it is for you step by step with a little bit more detail. So you've got the transport vesicle, right? And this budded off from where? From the rough endoplasmic reticulum, comes into the cis face, fuses with the cis face. Those proteins get dumped into the Golgi apparatus, and then they get modified and sorted and eventually shipped out. So how does the flow of protein synthesis, modification, and shipping go? Well, you're going to start in the nucleus. And what are you going to make in the nucleus? You're going to take DNA, and you're going to make an RNA copy of it, an mRNA in particular, which stands for messenger RNA. That messenger RNA is going to go to the rough endoplasmic reticulum, where it's going to be read by ribosomes, and those ribosomes are going to produce a protein product. The protein product of those ribosomes are going to be shipped into transport vesicles, and remember these just kind of bud off from the endoplasmic reticulum. They contain proteins, and they go to the Golgi apparatus. The Golgi apparatus modifies them and ships them out in condensing vesicles, which then mature into secretory vesicles. And ultimately, these secretory vesicles go on to either other organelles or they go outside of the cell. Here you can see it in a little bit more detail. Rough endoplasmic reticulum, transport vesicles, Golgi body, modifies the proteins, and now it's shipping out the proteins into these condensing vesicles, and these eventually mature into secretory vesicles, which are released, more often than not, released outside the cell, but they can travel to other organelles. Big picture stuff. Proteins are incredibly important for the cell to make. They are the most abundant macromolecule in your body, and they perform a variety of different functions. The rough endoplasmic reticulum is going to be the site of most of your protein synthesis, along with the cytoplasm. Once those proteins are made, they're going to go to the Golgi apparatus for modification, and then they're going to be ready to be shipped to their ultimate destination. What else does the Golgi apparatus do? Well, it gives rise to lysosomes. Lysosomes are vesicles that bud off from the Golgi, and they contain enzymes. And these enzymes are involved in intracellular digestion of both food particles and pathogens. Think of Lysol. Lysol is a cleaning agent. Well, lysosomes, it's derived from the same root. And so it's kind of the cell's cleaning organelle, if you will. Vacuoles, these are membrane-bound sacs that contain particles that are either supposed to be digested, excreted, or stored for later. A phagosome is a special vacuole, and what it is it contains ingested material from outside of the cell, and when that phagosome comes into the cell, a lysosome fuses with it, forming a phagolysosome. Okay, so here's an example. This is phagocytosis, and we'll get pretty familiar with this when it comes to the immune system and how it fights against pathogens. So here we have a pathogen, a bacterium, and it has become attached to these evaginations of the cell. These evaginations kind of come together up here and engulf that bacterium, and now we call this a phagosome. You can see a lysosome right here comes in and fuses with the phagosome, forming a phagolysosome. Those enzymes inside of the lysosome digest that bacterium, killing it. And then those digested products are going to be released from the cell. Okay, the next organelles we're going to talk about are the mitochondria, and these are very important organelles because they are going to be the site of energy production and storage. They are where ATP is made, and ATP is the energy currency of the cell. Mitochondria have two membranes, an outer membrane and then an inner kind of folded membrane. Those folds are called cristae, and the reason they exist is to increase the surface area of the inner membrane. And why you want to do that is because you want to pack as many enzymes into that inner membrane as you can, because the cristae membranes hold the enzymes and the electron carriers of aerobic respiration. Remember, aerobic respiration is where we basically take oxygen and sugar and turn it into ATP. So you want to have as many enzymes as possible to produce as much ATP as possible. Now, the mitochondria have their own DNA, and they're going to divide independently of the rest of the cell. And this makes sense if you think about it in light of the endosymbiotic theory. If you think way back evolutionarily, the mitochondria were once bacteria that were trapped inside of another prokaryote. And they evolved inside of that prokaryote and eventually evolved to the point where they could not exist outside of the other cell. That would be an early example of a eukaryote. DNA and prokaryotic ribosomes are going to be contained in the spaces around the cristae, and the space is called the matrix. So here you have a mitochondrion. You can see the outer membrane here, and then the inner membrane, all of these folds called cristae on the inner membrane. Within the inner membrane is the mitochondrial matrix, and within the matrix you'll find the mitochondrial DNA. Chloroplasts. So these are going to be found in plants, but also algae, and that's why we talk about them in microbiology. They convert the energy of sunlight into chemical energy through the process of photosynthesis. The chloroplast is going to have an outer membrane that covers an inner membrane, and that inner membrane is folded into sacs called thylakoids, and those thylakoids are stacked kind of like coins, and those stacks of coins are called grana. They have pigments such as chlorophyll. Chloroplasts are going to be the primary producers of organic nutrients for other organisms. Organisms that use chloroplasts are going to form the base of the food chain. Okay, so you can see the flattened sacs and the thylakoids, and you can see how they're stacked kind of like a tower of coins. Each stack is called a granum, plural grana. Okay, ribosomes. Ribosomes are organelles as well. They are not membrane-bound organelles, though. They're going to be composed of ribosomal RNA and proteins. They are found throughout the cytoplasm or embedded in that rough endoplasmic reticulum, remember? So where is protein going to be made in the cell? Well, it's going to be made primarily in the rough endoplasmic reticulum and then in the cytoplasm. Eukaryotic ribosomes, you will need to know, are larger than prokaryotic ribosomes. But like the prokaryotic ribosomes, they function in protein synthesis. Here we've got that close-up of the rough endoplasmic reticulum again, showing our strand of mRNA that's being read by 3 different ribosomes all at once, each one of them putting together a strand of the protein that the RNA is basically the recipe for. Okay, finally, you have the cytoskeleton, and this is going to be a flexible framework of protein strands, okay? Proteins, microfilaments, and microtubules. And what all of these fibers do is they anchor the organelles. They help move the cytoplasm around, such as during cell division. They help in locomotion through amoeboid motion. They help with cellular transport, and they also provide structural support. Here's the different fibers of the cytoskeleton. You have from smallest to largest actin filaments, then intermediate filaments, and then microtubules here. Okay, question. The eukaryotic organelle that is responsible for transporting vesicles inside the cells is the... Well, remember what organelle is going to be packaging things into vesicles to be sent off to other organelles or outside the cell? Well, that's going to be the Golgi apparatus. Okay, so let's talk a little bit about the different eukaryotic microorganisms. We're going to discuss fungi. I'll touch a little bit on algae, won't go into that in too much depth. We'll talk about protozoa, and we'll talk about helminths, or parasitic worms. Okay, so let's start with fungi. There is a lot of information in this PowerPoint about fungi. I'm going to try to just boil it down to the simplest concepts and what you need to know kind of as far as what I'm going to test you over. So fungi, the majority of these are going to be unicellular or colonial. There's a bunch of different species divided into 2 groups. So you've got macroscopic fungi, right? So this is going to be mushrooms, puffballs, things like that. And then you've got the microscopic fungi. This is what we care about. And there's 2 forms that microscopic fungi can take. It can take the form of hyphae. This is going to be long filamentous fungi. So it kind of looks like little filaments. And these are called molds. Or it can take the form of yeast. And this is a single-celled organism. It's round in shape, and it undergoes asexual reproduction. Some fungi can exist as both a mold and a yeast, depending on the environment. These are called dimorphic fungi, and you'll see that often in the fungi that are also pathogens. Okay, all fungi are going to be heterotrophic, meaning that they get their nutrients from another organism. The majority are going to be saprobes, and what a saprobe is something that gets its nutrition off of dead or decaying matter. So a majority of mushrooms are going to spring out of things like dead and decaying plants and animals. Some fungi are parasites that can live on the tissues of other organisms, but none of them are obligate parasites. That means they're more opportunistic. Like if the opportunity is there, sure, they'll set up shop and infect the tissue, but generally speaking, they are not harmful. We call a fungal infection a mycosis. Plural is mycoses. And you'll see that most fungal infections are superficial in nature, usually only involve the outer part of your skin. However, when a fungi do get into the tissues and into the bloodstream in particular, it can be extremely difficult to treat. Okay, the appearance of the different fungi under the microscope. So molds, just looking at them with your eyes, they're going to look cottony, velvety, kind of hairy or furry. And this is because there's a woven mass of hyphae, and we call this mass of hyphae a mycelium. Yeast, on the other hand, are going to form something called a pseudohypha, plural pseudohyphae, and I'll show you what this looks like in a minute, but it's a chain of yeast cells, and basically the yeast buds off from the cell. That's how they reproduce, so by forming a bud. And instead of totally budding off, some buds remain attached, and they kind of form a row or a chain. And it looks almost like a hypha, but it is not. Okay, so this is what hyphae look like zoomed way in. And you can see they're kind of like strands of hair. Molds, or the filamentous fungi, are going to look more like this on gross appearance. So you can see there's kind of this fuzzy, cottony appearance to them. And this is because of those mycelia. Singular is mycelium, and that's that tangled mass of hyphae. Okay? So that's why they look so furry. Yeast, on the other hand, these are going to be distinguished by their round to oval shape, and they reproduce asexually. They have a cell wall. They don't have any locomotor organelles, and they grow swellings on the surface called buds, which are then going to become separate cells. Now, here you can see we have a yeast cell, and you can see the nucleus is splitting. So you're getting some DNA in each part. The bud over here, which is starting to bud off, and the original yeast cell. And then you're going to have the bud attached. And if the next bud remains attached, and so on and so forth, you get this chain of yeast cells called pseudohyphae. Okay, fungal reproduction. Just to simplify, it's really complicated. But you can look at it kind of as one of two things. It's either going to be asexual, like the yeast cells do, or it's going to be sexual. Primarily, it's going to be through spores on reproductive hyphae. So primarily, it's going to be sexual reproduction. And the way fungi do this is they produce spores, and there's a couple of different ways that spores can be produced. You can see there's some structural differences here between these two spore-releasing mechanisms. And these are an example of the asexual spore formation, but you also have sexual spore formation. All right, so you can have asexual or sexual reproduction with fungi. Classification of the fungi is going to be based on the type of sexual reproduction, or if they are asexual, they would be classified down here. So identifying a fungus, we have special media that we use that is basically tailor-made for fungal nutrition. So it'll grow fungi on it very well and will not tend to grow bacteria or other microorganisms. So you would inoculate that media, and then you would grow the organisms and look for macroscopic and microscopic signs of asexual spore-forming structures, what type of hyphae they have, colony texture and color, physiologic characteristics. So, for instance, does it metabolize hydrogen peroxide? And then finally, you could look at the genetic makeup. Okay, there's a lot of information here. Most of it you don't have to worry about. Just know that in this phylum, there are very common molds, like bread molds, okay, like rhizopus, mucor. These molds are everywhere in the environment. Now, usually, if you're healthy, they don't bother you. But if you have severe immune deficiency of some kind, then you are at risk of developing an infection from these fungi. For example, mucormycosis is an infection of the typically the sinus cavity, but it can infect the eye and surrounding areas in patients with poorly controlled diabetes. Essentially, this otherwise totally harmless mold that's just floating around in the air, it basically eats the face in this infection. All right, phylum 2 has several notable species. In particular, it has Penicillium. This genus is one source of antibiotics. Particularly, this is where we get penicillin from. Aspergillus. This is a common airborne mold, can cause respiratory infections, and it's also toxic. Pneumocystis jirovecii. This is, unfortunately, a common cause of pneumonia in patients with AIDS. You've got histoplasma, the cause of Ohio Valley fever or histoplasmosis. Trichophyton, this is one cause of ringworm. Coccidioides immitis, this is the cause of another febrile illness. Candida albicans, this is the cause of the common yeast infection. Next you have phylum 3, which is going to include one human pathogen, Cryptococcus neoformans. Now, Cryptococcus is a yeast, and it can cause an invasive systemic infection. So it can get into your bloodstream and infect a bunch of different organs, including the skin, the brain, and the lungs. But classically, you see this as meningitis. And the textbook case is in a patient with severe immunodeficiency, such as an AIDS patient. So you'll see a common theme here is that fungal infections, severe fungal infections that is, are quite rare unless you are immunocompromised. Okay, fungi. There are good effects of fungi and there are adverse effects of fungi, okay? So the things that we don't like about fungi, they can cause mycoses. So infections. These infections usually are superficial and more obnoxious or ugly than they are dangerous. But they can cause severe systemic infections, especially in patients who do not have a strong immune system. You can have allergies to fungi, and some fungi produce toxins. Fungi are also destroyers of food, stored food, can mold, and they can also be plant pathogens. Beneficial impact, on the other hand, of fungi. They're a source of antibiotics, alcohol, vitamins. They decompose dead matter, and we use them when we make food and in genetic studies as well. All right, question. Which of the following is the primary characteristic of fungi? A, they are prokaryotic organisms. No, we're talking about eukaryotes. B, they perform photosynthesis. No, fungi do not photosynthesize. That is important to know, and it's important to know for the exam. Fungi do not perform photosynthesis. D, they have a cell wall made of peptidoglycan. They do have a cell wall, but remember, it's made up of chitin. So the answer is C. They are eukaryotic and heterotrophic. And you need to know this for the exam, okay? So not only are they eukaryotes, but they obtain their nutrition from other organisms. Okay, next are the protists. The protists are going to be any eukaryotic organism that is not a plant, an animal, or a fungus. Okay, so this is going to include your algae and your protozoa. Algae are photosynthetic organisms, so they have chloroplasts, which contain chlorophyll. They have a cell wall, and they may or may not be motile. They may or may not have flagella. You can see an algal cell here has 2 flagella. Okay, so algae, most of these are free-living, totally harmless to us. They are one of the constituents of plankton, and so they're going to provide the basis of the food web in most aquatic habitats. They're also going to produce a lot of atmospheric O2. All right, protozoa. This is a diverse group of organisms. They're all going to lack a cell wall. They're unicellular and mostly harmless. However, some protozoa are parasites that can be spread by insects. For example, the causative agent of malaria, which is Plasmodium. Okay, all protozoa are going to be heterotrophic, so they're going to acquire their nutrients from other organisms. They are not photosynthesizers. They lack chloroplasts. The majority of protozoa are going to be motile, so they have to have some sort of locomotor structure, so a flagellum, cilia, or pseudopods. Protozoa are going to have a life cycle with 2 distinct stages. So the motile feeding stage, or the, living its everyday life stage, is going to be called the trophozoite. However, when the environment becomes unfavorable, they can enter a dormant stage called a cyst. So here you can see an example of a protozoan. This is the trophozoite, or the active feeding stage. Here you have something happening, so it's the protozoan is drying. It's got a lack of nutrients. So what's going to happen is it's going to start to build a cyst. So the cyst wall kind of wraps around the organism and protects it from the unfavorable environment. Then when the environment becomes favorable again, such as moisture returns, nutrients are restored, the cyst wall is going to break open, and the trophozoite comes out and it's reactivated. Identifying protozoa. So they have a pretty unique appearance. So that allows for easy identification just based on their microscopic appearance alone. Here's one example. You can see all of these species are protozoa, and you can see how varied they are in their structures. So how do we identify a protozoan infection from a medical standpoint? Well, you're going to take a specimen, and then you're going to smear it directly onto a slide and observe it. You can do this either with or without special stains. I'll show you an example here. We have Giardia, the cause of giardiasis, which is a gastrointestinal infection. Amoebas are also protozoa. Entamoeba histolytica is the causative agent of amoebic dysentery. What happens in amoebic dysentery is the patient ingests cysts. Those cysts are going to get into the small intestine, and they're going to find a more favorable environment. So they're going to germinate, release that trophozoite, and that trophozoite is reactivated. The trophozoites are then going to migrate to the large intestine and grow, and from there they can invade several different organs, the liver, the lungs, and even the skin. Okay, question. Which of the following descriptions is true of both algae and protozoa? A, they are both photosynthetic. No, algae are, but protozoa are not. B, they are both always multicellular. No, they tend to be, they are unicellular. Algae, however, can be colonial. D, they are both heterotrophs. No, algae, they are photosynthesizers, so they can produce their own nutrients. Therefore, they are autotrophs. So the answer here is C. They both contain mitochondria. Your eukaryotic photosynthesizers, in addition to having chloroplasts, are also going to have mitochondria. Okay, parasitic worms or helminths. These are multicellular animals, so they're eukaryotes. They have organs for reproduction, digestion, movement, protection. Okay, so really well developed compared to the other microorganisms that we've talked about. These guys are parasites. They have mouth parts for attachment. Most have a well-developed reproductive tract. Okay, so essentially, in evolutionary terms, helminths have put a lot of their energy into reproduction. So they have very well-developed sex organs. They produce eggs and sperm. And fertilized eggs are going to go through a larval period, and this can be in or out of the host body. So the organism in which these worms undergo different developmental steps, that's going to be called the intermediate host. And the organism in which the worms reach adulthood and mate, that's going to be called the definitive host. And you need to know that definition for the exam. Okay, so let's talk about the major groups of helminths. You've got the flatworms and you've got the roundworms. And the flatworms are so named because they are flat. They don't have a definite body cavity. They're kind of rudimentary as far as their organ systems go. However, they have a very well developed reproductive system. You're going to have cestodes or tapeworms and trematodes or flukes. The tapeworms can have hundreds or thousands of segments. We call these proglottids. The flukes are going to be non-segmented. Roundworms, unlike the flatworms, are round, as the name suggests, but they also have a complete digestive tract. Okay, so this is an example of a tapeworm. This is its quote-unquote head, so the scolex. The scolex has hooks and suckers on it for attachment to the host. And then you've got these segments called the proglottids. And the proglottids are packed full of eggs. Okay, about 50 different species of worms parasitize us. These worms are distributed all over the world. Some are going to be restricted to some certain geographic regions. Generally, a higher incidence of helminth infections is seen in tropical areas. Most of these infections are going to be acquired through ingestion. So either ingesting larvae or eggs in food, in soil, in water, in just about any method. Some, however, are going to be carried by insect vectors, and some can directly penetrate the skin. And these infections afflict billions of humans worldwide. Okay, let's go over the life cycle of the pinworm, which is a very common helminth infection, especially among children. So it's going to start here when the child ingests an egg, a fertile egg. That egg is going to travel through the stomach. It survives the stomach and it's going to get into the small intestine. That egg is going to hatch. It's going to become larvae. The larvae are going to grow and develop. And then the male and female adults are going to mate. This generally happens in the large intestine. Now, when the female is done mating and she's ready to lay her eggs, she creeps down to the anus and she slips out of the anus and lays her eggs. This typically happens at night when the child is asleep. What happens is child feels intense itching at the site where the eggs are laid and reaches down there and scratches. Then the kiddo goes on, let's say, the next day to play with friends without washing their hands and passes along these eggs to their friend. Or let's say the eggs have gotten under her fingernails and she reinfects herself with the eggs. You can see that there's a lot of different ways that these eggs can spread, but the whole point is that the life cycle continues. It renews itself. So down here, when she spreads the eggs to her friend, now her friend is going to get a pinworm infection. Okay, question. Parasitic helminths reproduce by C, eggs and sperm. Remember, these are multicellular animals. So they are going to be, they're going to reproduce sexually using eggs and sperm.